The major regions of the brain are this big cerebral cortex up here that's organized into hemispheres or lobes. We'll come look at those in just a minute. Then we've got this cerebellum, kind of looks like a mini brain hanging out under the brain. You can see the brain stem here. And there's a fourth region deep in the brain called the diencephalon. So the four regions of our brain are the cerebral cortex, or the cerebrum, the diencephalon, the brain stem, and the cerebellum. So we'll talk about each of these in more detail as far as structures go today, and then for functions we'll talk about on Wednesday. So if we look at our brain, then from this picture we can see some of the different lobes that we need to worry about, and we can see other markings. So these ridges in the brain tissue, the singular is gyrus, plural is gyri, so all of these ridges are gyri, and we're going to be able to look at like our precentral gyrus here and know what it does, and the postcentral gyrus here and know what it does. So it's important to be aware that a gyrus is this ridge. All of these little dips down between our gyri are called sulci, or singular sulcus. So this central sulcus here is a pretty big marker. It's going to be what's splitting our frontal lobes from our parietal lobes, and we're going to use this to find our precentral gyrus and our postcentral gyrus. So central sulcus there in the middle. Another sulcus you can see here, the lateral sulcus, is separating our temporal lobe from the frontal lobes here, the parietal lobes here. So let's just talk about our lobes. The frontal lobes are found at the frontal region of the brain behind that frontal bone. Your parietal lobes are underneath your parietal bones back here. At the posterior part of your brain, we've got the occipital lobe. And then this region here behind your ears is called your temporal lobe. Your brain stem has actually three parts. The midbrain is this region right in here. The pons and the medulla oblongata. The brain stem is connecting the brain to the spinal cord. Your cerebellum is this region back here. It looks like a little mini brain. It's going to be important with coordinating motor output. And then your diencephalon is the last region we need to worry about. It's this kind of weird region in here. And it's got three parts. And it's not labeling one of them. I guess it is back here. The pineal gland is part of the diencephalon, the posterior aspect of the diencephalon. And then we've got this just weird kind of region here called the thalamus. That's your sensory relay station. And then your hypothalamus, found right here. And your hypothalamus is going to be your master gland. It's a master controller of the autonomic nervous system. So it's very, very important. And when you start 202, you're going to have to think about the hypothalamus, and it's going to be like the leader of all the rest of your glands that you're going to talk about. Hanging off of the diencephalon is the pituitary gland, but that's a gland. So you can talk about that next semester in 202. So those are our different regions overall and our markings. Let's look at these different parts of tissue that make up the brain. If we look at the brain, the superficial surface is full of gray matter. And the gray matter is made of the somas of our motor neurons or interneurons. Or interneurons, we'll say. So most of our gray matter is found superficial to our white matter, although we do have some regions of gray matter, clusters of gray matter, that are called cerebral nuclei. So cerebral nuclei are just these deep sections of gray matter that still have our somas of our interneurons. In that case, it'll be interneurons in here. Our motor neurons are only arising in the primary motor cortex. As you move down the brain, the cerebellum has the same arrangement with gray matter superficial to white matter, and then when you get down to the spinal cord, it actually flips, so that you've got white matter superficial to your gray matter. What is white matter? White matter looks white because myelin stains white, so it is white. So the white matter is the axons of our neurons. So we'll say myelinated axons. And interneurons. Somas of motor neurons and interneurons. All right, I know this is a long time ago, but back at the beginning of the semester, we talked about the organs in the ventral cavity being protected by serous membranes. And we said the organs in the dorsal cavity were protected by what? Meninges. And I said we'd revisit the meninges at the end of the semester. So here we are at the end of the semester, and meninges are going to be what's protecting the brain and spinal cord. And our meninges have 3 layers, 3 separate layers. The deepest layer is called pia mater. Pia mater is very thin, and it's going to dip down into the sulci. And this is areolar connective tissue. And it's holding the brain tight. Close to the brain, pia mater. Superficial to this, we've got what's called arachnoid matter. Arachnoid matter gets this name because it's these collagen and elastin fibers that arrange in this spiderwebby fashion. So there are these protein fibers that kind of arrange in this spiderwebby fashion. That's how it gets its name, arachnoid matter. And between the pia mater and arachnoid matter, we've got this subarachnoid space. And subarachnoid space is going to circulate cerebrospinal fluid. We're going to come back to cerebrospinal fluid in a minute. It's really important. But just be aware it's flowing through our subarachnoid space. So our most superficial layer of the meninges is called the dura mater. And it's got 2 layers. A meningeal layer that is hugging close to the arachnoid matter, and then a periosteal layer that's hugging close to the bone. So periosteal, peri around, osteum's always referring to bone. So the periosteal layer is around the bone, and this is dense irregular connective tissue. And there's a potential space in between these layers, but it's just a potential space. So the subdural space is that potential space here that you would only see if you were to like dry it out. So in the body it's not really full of anything. So don't say that the subdural space is full of CSF, because it's not. So those are the different layers of our meninges, and the meninges are going to help to protect the brain and the spinal cord from the bony structures that are protecting them. I mean, we're moving organisms, and our soft brain tissue would go beating into this bone if we didn't have the meninges there to protect it. So that's one way that we can help protect the brain, but then also we've got cerebrospinal fluid that helps as well. So we'll talk about that in a minute. These are what are called dural septa. I'm not going to really test you on these, but just be aware that these are extensions of dura mater that come down between the lobes of our brain and give some more support. What I am going to test you on, especially for their protective function, are the brain ventricles. So these are spaces in the brain that are full of cerebrospinal fluid. So we've got 4. You've got a left and right lateral ventricle. So these 2 big ventricles are your lateral ventricles. They are connected to each other through this interventricular foramen. And that interventricular foramen, it's in between the ventricles. So the interventricular foramen comes down and drops the cerebrospinal fluid into this 3rd ventricle. So this is the 3rd ventricle. And then the 3rd ventricle is connected to the 4th ventricle by this stalk here called the cerebral aqueduct. So this is a testable model. You've got right and left lateral ventricles that are connected to each other by the interventricular foramen. That's also how they're connected to the 3rd ventricle. And then the 3rd ventricle is connected to the 4th ventricle by the cerebral aqueduct. So make sure that you're comfortable looking at this model. And our ventricles are just these spaces in the brain that are full of cerebrospinal fluid. Cerebrospinal fluid is very important. It serves some important functions for the brain. It gives it buoyancy. So buoyancy is like the ability to float. So the brain is floating basically in cerebrospinal fluid. And what this does is it lightens the apparent weight of the brain. Buoyancy is the first function. It provides this kind of ability for the brain to float and lightens the weight of the brain. The second function that it does is it gives protection. How does it do this? It acts as a liquid cushion, and it slows the movement of the brain. So that as you're headbanging really fast, your brain doesn't go whacking into the bones. It slows the movement of the brain. The last thing it's going to do is provide some environmental stability. So it can exchange nutrients and waste with the interstitial fluid. And it can help to maintain normal ion concentrations of that interstitial fluid. So it's going to help to provide some environmental stability by ensuring that we're keeping the composition of our interstitial fluid pretty constant and keeping the composition of our CSF pretty constant. So we'll exchange nutrients and waste, and we'll manage ion concentrations. So cells are going to help with this, but CSF is going to act as this medium through which we can have this exchange of different materials from the blood and back through the CSF and then through the interstitial fluid. So how is CSF formed? We talked a little bit about choroid plexus when we were talking about brain tissue or nervous tissue, and that's because we've got these specialized glial cells and pendymal cells that are going to be found around capillaries. This structure is called the choroid plexus, and it's specialized. This is part of the brain that is lacking blood-brain barrier, and it needs to be lacking blood-brain barrier because these ependymal cells have to be able to take things from the blood to make the cerebral spinal fluid. So on our little ventricle model here, these little pink regions are choroid plexus. Are you actually going to be able to see choroid plexus with your eyes? No, it's microscopic. But this is kind of showing you where you'll find choroid plexus in the ventricles. And it's just this specialized region where we've got these ependymal cells associated with a capillary. And we need that because we need to be able to pull things out of the blood, like nutrients, like important ions we could be low on. We have to get our waste back out. So the choroid plexus is found in your ventricles and is going to produce our cerebral spinal fluid. So our cerebral spinal fluid is produced in your choroid plexus. We can see the way that it flows. It flows between our lateral ventricles in that interventricular foramen, and then it's going to flow down into our third ventricle, from our third ventricle to the fourth ventricle through the cerebral aqueduct, and then it's going to also come up into that subarachnoid space and go down into the central canal of the spinal cord. So that we've got CSF flowing through your brain and through your spinal cord, helping to keep the concentrations of our extracellular fluid in the way they need to be and all of that jazz. I'm not going to test you on the specifics of CSF flow, but I want you to know what's important for. Or if I were to ask you through here, you could describe cerebral spinal fluid through your ventricles. So if I posted this at a station and said, describe for me the production or the flow of cerebral spinal fluid through these structures from superior to inferior. And I would want you to say, well, it's produced in the choroid plexus of our lateral ventricles, travels through the interventricular foramen to the third ventricle, through the cerebral aqueduct to the fourth ventricle. And then we're done with this model. All right, any questions so far? All right, this is another thing we talked a little bit about last time, the blood-brain barrier. And the blood-brain barrier is formed by these astrocytes who stick these perivascular feet out and wrap around our capillaries. And they prevent the passive passage of materials from these capillaries into the blood, into the brain cerebral spinal fluid. So we talked about the important functions of the blood-brain barrier last time. It's mainly just to prevent things from just passively getting through here. So that anything that's getting into the brain, cellular fluid has to do so through our astrocytes. They're going to act as like this barrier and have some control over what gets into our brain. So the only places in the brain that are missing blood-brain barrier are places where we have to have contact with the blood. So we'll say it's absent in the choroid plexus, but it's also absent in the pineal gland, and it's absent in the hypothalamus. So our glands, our endocrine glands, release their product into the blood. So these are both endocrine glands. They've got to be able to have access to blood so they can release their product into the blood. All right, any questions so far? Then the last thing we're going to talk about today is we'll just go over the different regions of the brain, and then I'll let you look at brain models and work on your lab. And then Wednesday we'll finish up with our functions and our cranial nerves. So our brain we organize either into lobes or into hemispheres. So we've got a left cerebral hemisphere and a right cerebral hemisphere. They're separated from each other by the longitudinal fissure. So there is lateralization in 90% of the population, and the left cerebral hemisphere will be the hemisphere that's in charge of like all of the analytical things. So this tends to be the more analytical hemisphere. So problem solving, although that's really part of the frontal lobes as well, but it's really going to be what you're doing most of your like problem solving, your verbal, all of that's happening in the left side. In the right side, this is where like artistic, visuospatial things come in. So most people tend to have those activities being controlled primarily from the right cerebral hemisphere. Now you've always got your brain working on both hemispheres, and all of the lobes are working, doing a variety of things, but we can say in general there are some specializations. We'll look more at those on Wednesday as well. But that's kind of our start, the way that we look at our different hemispheres. The left is the really analytical one, the right's more artistic. If we look at these lobes and the function of these lobes, the frontal lobes, well, this specific area right here, this prefrontal or precentral gyrus, is in charge of our voluntary motor commands. So all of our voluntary motor commands are going to originate in the precentral gyrus. So that makes the frontal lobes really important for motor control. They also have these regions, the prefrontal cortex, that's going to be where your personality is found. So the frontal lobes are doing a lot of our higher order cognition. So your frontal lobes will do like problem solving, they'll do analysis. This is where we'll find our personality. So in that one of your last major neuronal clippings, when you're 25, you solidify all the connections to your prefrontal cortex. And that's really when you really get the ability to problem solve and analyze in a way that you kind of never were before. And the only people who've really caught on to that are the rental car companies, but they won't let you rent a car until you've made your connections to your prefrontal cortex. Other ways that you can think about what these prefrontal lobes are doing. I don't know if you've ever seen One Flew Over the Cuckoo's Nest. They used to do frontal those lobotomies and just go up through your nose and just like totally jack up your prefrontal cortex. And then after that, you like have no personality. So I mean, it was quite interesting. They realized probably not very cool, but there has been a resurgence in doing lobotomies. They just do it with lasers and things now. And some people who have like bipolar disorder and things are going and having their brains lasered. I don't know how I feel about that, but that's just something to be aware of. So your frontal lobes are really what make you you. If we look posterior to this central gyrus, we've got our parietal lobes and our central sulcus. This post-central gyrus is what's going to be responsible for our primary somatosensory cortex. So all of those senses that you're aware of on your body is coming in here. Not our special senses, these are our general senses. So this is our primary somatosensory, meaning sensory information that we're aware of. We'll say general sensory info that you're aware of is going to be mapped out on your postcentral gyrus. The rest of the parietal lobes are like it's a sensory association area. So your parietal lobes are really helping to process your sensory information. So we'll just say the rest is sensory association. And we'll talk more about the sensory pathways for the next test. Our occipital lobes are the most posterior lobes. This is your primary visual cortex. And then you can't see the temporal lobes here, but you can see them on your models. These guys are your temporal lobes. They're inferior to your parietal lobes. So right here, our temporal lobes are going to be important for our auditory information. So our auditory information comes in there. Olfaction, which is your sense of smell, is actually also processed in the temporal lobes.